In this video, we're going to talk about the enzymes, and we're going to talk about what enzymes are made up of, what are their roles in our body, and what are the different type of reaction enzyme can show. So now let's talk about the enzymes. Enzymes are proteins in nature that act as a biological catalyst. So enzymes are made up of proteins, as we already learned from the previous chapter, which is proteins, amino acid proteins. That enzymes are protein and they act as a biological catalyst. And what is catalyst mean? Catalyst are any substance that speed up the rate of a chemical reaction, right? So let's talk about why enzymes are being called as biological catalyst. So like I already told you, catalysts are any substance that speed up the chemical reaction, right? And why enzymes are called biological catalysts? Because they work in living organism, right? So enzyme catalyze nearly all chemical reaction taking place in the cells of a living organism or body. That's why we call as biological catalyst, and they catalyze the chemical reaction, or they speed up the biological chemical reaction by lowering down the activation energy of the reaction. What is the activation energy? Activation energy is the lowest amount of energy that any chemical reaction need to convert into a product or to give a product. So biological catalysts normally lower it down this requirement of energy. If you can see on this graph, y-axis is called energy, x-axis is the progress of the reaction. So this is the reaction, this one is uncatalyzed, means we don't have any catalyst in here. But the purple one is with enzyme. So if you can see how much energy is the enzyme lowered it down, how much activation energy is already lowered down by the help of enzyme, right? So enzyme increase the rate of a reaction by lowering down the activation energy. So now let's talk about what's special about the enzyme. So enzymes are highly specific, means they carry out a special or a specific chemical reaction that is designed for that particular enzyme. So there are different type of specificity. First one is absolute specificity. What does it mean by absolute specificity? So it means one particular enzyme act only on one particular substance or special substance, right? So each enzyme has its particular substance on which they can react, right? Relative specificity means enzyme act on structurally related substance. Even though the function of the substance are different, but if they are structurally looks like similar, enzyme can work on it if they have some common structural feature. And the third one is stereochemical specificity, means enzyme has the ability to distinguish between stereoisomers. If you have stereoisomers 1 and 2, then enzyme E can differentiate between 1 and 2 and then can just catalyze the reaction for that one particular form of stereoisomer, but not the second one, because they can easily detect which type of stereoisomer I want to do a reaction or I want to participate in, right? So for example, for the specificity, one example is urease. As the name is saying, it act on urea in the presence of water and convert urea into carbon dioxide and ammonia, right? So the name is urease, so it always react with urea or help in the speed up of a chemical reaction where urea is being hydrolyzed. So now let's talk about the enzyme cofactor. But before we talk about enzyme cofactor, what's the special about the enzyme? Like I already told you, enzymes are biological catalysts. They speed up the chemical reaction by lowering down the chemical or activation energy Important thing is enzyme doesn't get consumed during the reaction. Means if the enzyme is being utilized during a chemical reaction, at the end of the reaction you will get the same enzyme. So it don't get consumed during the reaction. That's the important thing. Okay. So now let's talk about the enzyme cofactor. What are cofactor means? So cofactor are any non-protein molecules or ions required by enzyme for its catalytic activity. So that means if enzyme wants to do some biological activity or biological function, some non-protein molecule has to get or ions has to get attached with the enzyme before enzyme can perform its biological activity. Means let's assume this is enzyme 1E, and if this enzyme has to work on a reaction A is to B, this enzyme is not going to work on this, or A is not going to convert into B without this enzyme. So even though the enzyme is present, enzyme cannot participate or load it down the chemical or speed up the chemical reaction of converting A to B. Why? Because it needs one cofactor, which is a non-protein molecule or ion. So that means enzyme need to get attached with some cofactor, and once the cofactor get attached with the enzyme become active, and then it catalyze the reaction of A to B, right, or speed up the chemical reaction. So so cofactor can be you can divide cofactor into two parts, either as coenzyme or inorganic ions. So cofactor can be of two types, coenzyme and inorganic ion. So what is coenzyme? Coenzymes are those cofactors that are organic in nature, and this organic cofactor is normally formed by the body through vitamins, right? So mostly organic cofactors are formed by vitamins within the body, right? And inorganic ions are some metal ions such as magnesium and zinc and iron. So they are some metal ions. So you have to know the difference. Coenzymes are organic in nature, whereas inorganic ions, as the name is also inorganic and made up of metals. So some enzyme need metals to carry out the function, like, right, and some enzyme need some organic cofactor that is derived from vitamins. And there is one more term called apoenzyme. So apoenzyme is catalytically inactive enzyme. Means enzyme is there, but they are not active, right? They are not doing anything. Even though they are present, they are not helping the reaction to speed up, right? Unless or until some cofactor is attached to this enzyme. If some cofactor attached with this enzyme become active and then help in the speed up of the chemical reaction. So apoenzyme is an inactive enzyme but once apoenzyme binds with the cofactor, it becomes active enzyme, right? So apoenzyme plus cofactor is an active enzyme. So make sure you know all this terminology, coenzyme, apoenzyme. This is also important. So now let's talk about how we name any enzyme. So we have different ways how we name an enzyme. So we normally name an enzyme by replacing the end of the name of the reaction or the reacting compound. So in which reaction that particular enzyme is being used, or on which compound that enzyme is being used, right? You have to use that name and end with ASC with a. For example, sucrase. As we know, this enzyme work on sucrose, because the first term normally says sucrase, and then you have ASC, sucrase. That means this enzyme work on a molecule called sucrose. Similarly, if an enzyme work on one particular reaction, then you have to use that reaction and end the word with ASC. For example, oxidation reaction. So if any enzyme is helping in oxidation reaction, the name of that enzyme will be oxidase, right? So the name is always end with ase. So we have couple more example. Enzymes are classified by the reaction they catalyze, like we already learned in the previous slide. So there are mainly 6 main classes of enzyme, or 6 main reaction that enzyme helps in. One is oxidation reduction. So the name of the enzyme is oxidoreductase. If you have any transfer of groups of atom in during the reaction, if you're transferring the group with the help of enzyme, that enzyme is called as transferase. You are transferring it. If you are doing hydrolysis, means in the presence of water you are breaking a bond, then hydrolase. Lyase is that enzyme that add or remove atom or from a double bond. Then you have isomerase, means you rearrange atom to make isomers. Then you have ligase. Ligase is a special enzyme that we're gonna learn in the last chapter in carbohydrate metabolism, that use ATP to combine its small volume molecule, and then we're gonna learn about ATP over there too, right? So you can use right now ATP is the energy molecule. Okay, so now let's try to do one problem and see what we learned till now. Match the type of reaction catalyzed with an enzyme class. So the first one is form bond between molecule using ATP. So the time I see ATP energy, we know this is ligase. Second is rearrange atom in a molecule to form isomer. The time I see isomer, I know this is isomerase. Third one is transfer a group between two compounds. If you're transferring a group, that is transferase. And the fourth one is hydrolysis reaction, hydrolase. These are the solution that we already discussed about. What are the different enzyme belongs to which of the option. Right? Now let's talk about some special area on the enzyme that normally helps in the reaction. Right? So till now we just learned what enzymes helps in to speed up the chemical or biological reaction within the body. But how the enzyme normally speed up the reaction, we're gonna see in this slide or on this topic, which is active site. We're gonna learn what is active site on enzyme. So active site is present on the surface of an enzyme, and it is a small region that binds the substrate and catalyzes the reaction of that substrate. What does it mean? If this is an enzyme, so on enzyme surface you will have a small area which is called as active site, and on this active site substrate is going to bind. Substrate binds, right? So active site is very important, which is present on the enzyme, and through this site or this area enzyme binds the substrate and converts substrate into a product, right? So you will see on this structure over here, this is an enzyme and this area is your active site, and if you can see, this is the substrate, and this substrate is bound to the enzyme through some kind of interaction like hydrogen bond, hydrophobic interaction, salt bridges. So because of all this interaction, substrate can bind to the enzyme active site where all the reaction normally happens, right? So what is active site? Active site is a region with an enzyme, I will talk about it, that fits the shape of the reacting molecule called substrate, and it contains amino group R group that binds to the substrate and release product when the reaction is complete. So what does it mean? So this is an enzyme, this is active site, where the substrate normally bind. So let's assume this is a substrate. The substrate bound to the enzyme through some kind of interaction that holds the substrate within the active site. And then after the substrate is bound to the active site, enzyme does its function, its biological activity, and then after that you're gonna see again enzyme plus you will see a product. Right, so the substrate is converted into a product and you have now active free site again. Right, so this is, and then what's the name of the complex that you have? Substrate plus enzyme. So substrate plus enzyme we call as enzyme substrate complex, ES complex. E means enzyme, S means substrate complex. So whenever the substrate bound to an enzyme on the active site, they form a special complex called ES complex. And after the reaction is done, you will see enzyme is free again, and then the substrate is going to convert into a product. Right. And now this enzyme again bind to another substrate. Right. So again, this is an enzyme catalyzed reaction. That's the same thing that I already explained to you. This is an active site. You have the substrate in here, and this substrate is bind through some kind of interaction to the enzyme because they need some kind of interaction. All the substrate will come out from the enzyme active site, so it has to bound to the enzyme through some kind of interaction. Right. Then let's see how this enzyme reaction goes. First you have an empty enzyme with an empty active site. Now the substrate come close together. Substrate goes to the enzyme and bound to it to form enzyme substrate complex. Enzyme substrate complex. And once the complex is formed, enzyme does its activity and converts substrate to a product, and enzyme just come out without being used to it. Now this enzyme is again ready to make or combine with another substrate. Right. So this is normally the basic sequence how the enzyme normally catalyze the reaction with the help of active site. Right. So let's take one example of the hydrolysis of disaccharide called lactose. We're gonna see an example where the lactose is a substrate, and if it's a lactose, then what's the name of the enzyme that do the hydrolysis of lactose? Lactase, right? As the name is saying, is a lactase. So we know lactose is a disaccharide, right? And lactose is form of two. If it's a disaccharide, I know this is formed between galactose, two monosaccharide, right? Galactose plus glucose through a glycosidic bond, right? So how does this lactase normally catalyze the reaction and convert lactose into its component monosaccharides, which is galactose and glucose? Let's see over here. So you have an enzyme, you have substrate which is lactose, enzyme is lactase. So once the substrate bind to the enzyme through an active site, they form ES complex, means lactase enzyme substrate lactose complex, ES complex. And once the complex is formed, enzyme is going to break the glycosidic bond between the lactose, and they're gonna form two products, glucose and galactose. Because once the bond is broken between galactose and glucose, then you're gonna get two monosaccharide union, and now the enzyme is again empty, right, with an active site, and now is ready to attach with another lactose molecule, and that reaction keep on going on, right? So this is how enzyme normally works. So now we're going to talk about the enzyme action, how the enzyme normally function within the body. So there are two models that normally tells about the enzyme action, how the enzyme work with the substrate. One is lock and key model, and the second one is induced-fit model. So we're going to talk about this individually though. Now let's talk about the first one is lock and key model. So in lock and key model, people assume or scientists assume active site has a rigid, non-flexible shape. Means, let's say if this is an enzyme and this has some kind of squarish, this is your active site. So according to this model, this active site is non-flexible. So if this is the shape of the active site, it remains the way it is. So if any substrate that can fit, only, let's say if I have two substrate, one is square and one is, it's a hexagonal, this is two substrate, then what's the best substrate that can fit into the active site? This is the square one, because it can easily attach, goes into the active site and then bind to the active site, whereas hexagonal shape is very difficult to just accommodate within the active site. So according to this model, enzyme binds only to the substrate that exactly fit the active site, right? Because the active site doesn't show any flexibility to accommodate any other shape of the substrate, right? That's the main thing about this lock and key model. And then it says substrate is the key that fit the lock. So for example, for every lock we have only one key, right? One special key is made for one special lock, right? So you cannot have multiple key for the one lock, right? Similarly, in lock and key model, substrate is like a key that can fit that particular lock. Lock is an enzyme. Like you have only one key for one lock, similarly you can have only one substrate, one type of substrate for each enzyme. And this model was a static one that did not include the flexibility of the tertiary shape of enzyme. So the enzyme, the active form of an enzyme is always a tertiary structure. And we already talk about enzymes are proteins, and we already talk about the different structure of protein, primary structure, secondary and tertiary structure. So that means that most of the enzyme have an active form when they occur as a tertiary structure, when they're present in the form of tertiary structure of protein, right? So this is important. You need to make sure if somebody talk about enzyme, we are talking about most probably the tertiary structure of the protein, which is an active form of the enzyme. So in the previous slide, we talk about the lock and key model, where we talk about the active site of the enzyme is fixed, is not flexible. So only that particular substrate can get into the active site, which the active site can accommodate, right? With the structure is, the shape of the substrate should be similar to the shape of the active site, right? But the second model of the enzyme action, which is called Induced-Fit model, as the name is already saying, Induced-Fit means the active site of the enzyme is very flexible and is going to fit any substrate, and or is going to change according to the substrate that enzyme is being catalyzed, right? So in Induced-Fit model, enzyme structure is flexible. This is important. Whereas in lock and key, enzyme structure is fixed, or the active site is fixed, not non-flexible, and is not rigid and adjusts to the shape of the active site in order to bind to the substrate. So this is the difference between the two models. Here the active site shape can be flexible and can adjust according to the shape of the coming substrate, right? And because of this, the range of the substrate specificity increases, right? And the shape change improve catalysis during reaction. So for example, this is your enzyme, and again you have this is your active site. So this active site has a shape of something squarish, right? So if this enzyme is close to a substrate which is not squarish, but let's say this is circular, this is your substrate. So if the enzyme comes to the substrate, the active site shape is going to change a little bit to accommodate this substrate now is some kind of semicircular, right? Now if you have a substrate, same substrate, it can go and directly fit inside it. So what happened over here, induced fit model, according to the induced fit model, the active site of the enzyme is accommodating the shape of the substrate. That means it adjusts according to the shape of the substrate, right? So depending, if you have a circle, now if you have, let's say, again a square, after this reaction the enzyme can again adjust its shape to the square so that this substrate can go inside it, right? So that means the range of substrate specificity also increases. Means first enzyme is working on the circular substrate, again second time it's gonna work on the squarish substrate, so they can catalyze many substrate with different structure or shape, right? Only one enzyme can do multiple reaction with different shape of the substrate. So this is an example. If you can see over here, this is a substrate, for example lactose, and if you can see, this is hexagonal again, but the active site is some kind of triangle. So is it not possible to put this substrate into an enzyme which is lactase, because we are doing lactose? But because of the induced fit model, the active site is going to adjust according to the shape of the substrate and then adjust the substrate in its active site. Now you will see, now the active site is not some kind of triangular or like this kind of structure or shape, right? And then the enzyme substrate complex form and the bond is going to break, and then you're gonna form galactose and glucose. So this is an example of induced fit model where the enzyme change its active site shape according to the shape of the substrate. Now let's talk about couple of the questions that apply all the concept that we already learned from the previous slide. What is active site? Active site is a section of the enzyme. This is not complete enzyme but a part of the enzyme, or a small area on the enzyme, right? So this is your answer for the first one. Second one, in the induced fit model, the shape of the enzyme when substrate bind stays the same or adapt to the shape of the substrate. Means the correct answer is it adapt according to the shape of the substrate. That's why it says induced fit model. Substrate induce the enzyme to adjust according to its shape. So that's your correct answer, B, option B. Right, so this is a solution already given, so we already talk about this thing. Now we're gonna talk about the enzyme activity. What is meant by enzyme activity? Enzyme activity is the rate at which an enzyme catalyzes a reaction. How fast the enzyme is catalyzing the reaction. And there is a way how can we calculate the rate of enzyme activity. We normally calculate by using a number called turnover number. What is turnover number? Turnover number is the number of substrate molecule acted on by one enzyme molecule per minute. Means how many substrate molecule is converting into a product by one enzyme per minute. Means if you are producing high number of product per unit time, means the activity of that particular enzyme is higher compared to that one enzyme that producing less amount of or lower amount of product within that within that 1 minute, right? For example, if this is enzyme 1, this is enzyme 2, this is substrate 1, this is substrate 2. If enzyme 1 convert this substrate into a product, let's say if this enzyme work on 200 molecules of substrate and then convert all these 200 molecule into a product, whereas enzyme 2 within 1 minute. If this enzyme to work with a different substrate and it can just work on 100 molecular substrate too, that means it can just make 100 product within the same 1 minute. So that means the activity of enzyme 1 is greater than activity of enzyme 2, or you can say the turnover number of enzyme 1 is higher than turnover number of enzyme 2. So this is how we normally calculate the rate of the enzyme or enzymatic activity. There are certain factors that can affect enzyme activity, and then we're going to talk about all these factors. What are the factors that can affect the rate of a biological reaction? The first one is enzyme concentration. So what's going to happen if we increase the enzyme? So let's assume we have enough substrate, and we are not running out of a substrate. If we keep on increasing the enzyme number, that means you are increasing the number of active site, because each enzyme has one active site. So if you're increasing the number of enzyme into the solution, means you are increasing the number of active site. If you're increasing the number of active site, means you're increasing the formation of enzyme substrate complex, right? Because each active site binds with one substrate. If you have two active site, it's gonna bind with one more substrate, right? So similarly, if you're increasing the enzyme concentration, you're increasing the active site, and if you're increasing the active site, you're making more enzyme substrate complex. Means enzymes are working on multiple substrate that is already present into a solution. If they are making a complex, that means they are converting substrate into a product. So the final word is, if you increase the concentration of enzyme, you are increasing the rate of a chemical reaction. Means you are providing more enzyme to the substrate to get converting into a product, right? So that means rate of a reaction is directly proportional to the enzyme concentration. So if you see this graph, y-axis is rate, x-axis is your enzyme concentration. So that means this is a straight line, is a directly proportional. So I can say rate is directly proportional to the enzyme concentration, right? Second is your substrate concentration. How you can affect the enzyme activity if you change the substrate concentration. If the enzyme concentration is constant, if enzyme concentration is constant, is not changing. So assume if I have, let's say if I have 5 enzyme, 1, 2, 3, 4, 5 enzyme, right? 1, 2, 3, 4, 5. So we cannot increase the number of enzyme. In this particular scenario, we have just constant number, which is 5. And if I keep on increasing the substrate concentration, let's say if I put first substrate, it's gonna bind with this enzyme 1 and converting into a product, right? So the rate increase. If I put second substrate molecule, is gonna bind with the second. If I put the third and keep on adding the substrate, is gonna bind with the enzyme and then converting into a product. So that means till I put the fifth substrate, the rate is directly proportional to the enzyme or the substrate concentration, because the number of substrate that you are putting into a solution, they keep on reacting with the enzyme, with the free enzyme. But the time I put sixth substrate, we don't have any more enzyme that this substrate can go and bind to it, right? Because we had just 5 enzymes, and all these 5 enzymes are working right now to convert all the 5 substrate into a product. So after some time, the rate of a reaction become constant. If you see this region, the rate is not gonna increase. So every minute or every time, you always gonna get only 5 product at one particular time. Or if you put the 6th substrate, you're gonna wait some time until each and every enzyme is done with the chemical reaction, and again it's ready to perform one more reaction with the sixth substrate. So that means once for the first couple of for some particular time the rate is constant or rate is directly proportional to the substrate concentration, means the number of substrate you are increasing, the rate increasing. But after some time the rate will max out because you don't have any more enzyme. It doesn't matter how much substrate you're gonna add it, the rate doesn't change, so they become constant, right? And this maximum rate we call as Vmax, means maximum rate. So maximum rate is that concentration of the substrate that will give you a maximum efficiency for the enzyme, maximum catalytic ability of the enzyme, right? Let's talk about the third factor that affects the enzyme activity, which is temperature. So we're going to talk about how temperature affects the enzymatic activity. As we know, most of the chemical reaction normally increase with increasing temperature. Same thing apply to here. The rate of enzyme catalyzed reaction also increase with temperature, but you cannot keep on increasing temperature because enzymes are proteins, and if you keep on increasing the temperature of the enzyme catalyzed reaction, enzyme is going to denature. Why? Because we already talk about higher temperature can denature the protein because it can disturb the interaction that keep the tertiary structure of the protein stable, right? And we know enzymes are tertiary structure of protein. They are proteinous nature. So each enzyme has its optimum temperature where they can work best, right? Or with a higher efficiency. So temperature with a range of 25 to 40 degrees Celsius, we normally see a highest enzymatic activity. After that, the enzyme is going to denature. So between 25 to 40 degrees Celsius, you will see highest enzyme activity. The reaction rate is going to be lower, below or above than this, because above than 40 degrees Celsius it's going to denature, and below than 25 degrees Celsius the enzyme cannot work, right? Because they need some kind of energy. Beyond this temperature, enzymes are vulnerable to denaturation. Like I already told you, a higher temperature sometimes denature the protein or means enzyme. So if you can see, if this is x-axis is your temperature, y-axis is your rate. So the first couple of temperature, let's say from this start was 0, if you keep on increasing the temperature, the rate is increasing, but after some time the rate is going to decrease because the protein is become denatured or the enzyme become denatured after that particular time. So this is the optimum temperature at which you have highest rate, right? So this is the optimum temperature. So make sure you know this thing, right? What's the optimum temperature? So each enzyme has its own optimum temperature. The fourth one is your pH. How does the pH normally affect the enzymatic activity? So enzymes are most effective in a narrow pH range. So they are more efficient in a small pH range, and its lack active pH value lower and higher than the optimum pH. So this narrow pH we call as optimum range. Optimum range where the enzyme can work, and above and below that optimum range the enzyme cannot work, it becomes denatured, right? Because each enzyme needs its own optimum pH to work, right? And in biological organism is usually pH around 7, close to 7, right? So if you see from the graph, y-axis is your rate again, x-axis is your pH. If you see, if you keep on increasing the pH, the reaction rate increases, or the enzymatic activity increases. But after certain pH where you have the highest activity or highest rate, After that particular pH, if you keep on increasing, the activity of the enzyme is getting decreased. Why? Because enzyme normally denature if you keep on changing the pH range very much, right, in a higher range. So because of the denaturation, that enzyme activity also decreases because enzyme normally become dead. So this is some example of the optimum pH value. So one of the enzyme is pepsin. It present in stomach, right? And then the pH of the optimum pH for this pepsin is 1.5 and 2.0 because the pH of the stomach is acidic, right? So we have different examples. So this will tell you different enzyme has its optimum pH, right? So if, for example, if you put this pepsin at a pH of 8, this gonna denature because what's its optimum pH? 1.5 and 2.0, which is acidic. But if you put this pepsin in a pH 8, it's gonna be denatured, and then you won't see any catalytic or enzymatic activity for this enzyme at pH 8, right? So now let's talk about a question and then apply the concept. Sucrase has an optimum pH of 37. So that enzyme sucrase has an optimum pH of 37 and an optimum pH of 6.2. Determine the impact of each change on the enzyme activity, whether it's no change, increase or decrease. So the first option is, if I change or if we change the pH to 4, what's going to happen to the enzymatic activity if I put sucrase at a pH of 4? Because we know the optimum pH of sucrase is 6.2 and now we are putting at a pH 4, that means that is below the optimum pH. That means the activity is gonna decrease because each enzyme has optimum pH to work, where if they can show its highest activity. Option B is running the reaction at 70 degrees Celsius. So now the temperature we are using is 70, but what's the optimum temperature for this enzyme? 37. So that means the reaction rate will also decrease because at that high temperature enzyme is gonna denature, and then if the enzyme become non-functional, they cannot show any efficiency, right? They cannot do any reaction. So that means the reaction rate will decrease. Right. So this is the answer that we already talked about. Now we're going to talk about the inhibition of the enzyme, how we can inhibit the function of the enzyme or catalytic efficiency of the enzyme. So there are some molecules that can do this work to inhibit the activity of the enzyme. We call that molecules or substance as inhibitors. So inhibitors are molecules that cause loss of catalytic activity of the enzyme. And it also prevents substrate from fitting into the active site. It prevents substrate from fitting into the active site. What does it mean? Let's say if this is your enzyme and this is your substrate that should go into your active site, but let's say if you have the same shape of the inhibitor, I is your inhibitor. So instead of substrate, if inhibitor goes to the active site first, it's going to bind to the active site, and then the substrate cannot enter it. If the substrate cannot enter to the active site, enzyme cannot produce a product. So that means the efficiency of the enzyme decreases because inhibitor already bound to the active site and already covered the active site. So for example, that is already given in the reaction that is given at the bottom. Enzyme, if enzyme is present, if substrate is present, they're gonna form enzyme substrate complex, and then after that you're gonna get enzyme and product. But if the inhibitor is present and if it binds to the enzyme, then you're gonna get enzyme inhibitor complex. And once this complex form, then this enzyme is no longer available for the substrate to bind to, and then if there is no substrate bound to the enzyme, then you will not get any product. right? So no product. So why we need to study about the enzyme inhibitors? Why we need to study about? So reason to understand enzyme inhibition is because most of the medicine that we usually take and most of the poison that normally function as an inhibitor. Because if an enzyme we don't want to work in our body, or which we don't want, or the body doesn't want that enzyme to do its function, they normally inhibit that enzyme with the help of inhibitor, and this inhibitor can be in medicine or poison, and that will decrease the rate of a catalyzed reaction. And some substance found in our cell also inhibit the enzyme catalyzed reaction, because in our body we need internal regulation. We don't want every time the enzyme, even though the enzyme we don't want any product, but the enzyme is keep on producing the product we don't want in our body, so that by body or also self-regulate its enzymatic action by producing an inhibitor which can inhibit the function of the enzyme, so that the enzyme catalytic reaction is an internal regulation, right? So there are two types of inhibition. One is called reversible inhibition, second one is irreversible inhibition. As the name is already suggesting, your reversible inhibition means this inhibitors or inhibition can be reversed by some means, whereas irreversible inhibitors means once the inhibitor binds to the enzyme, you cannot do anything to reverse that reaction. The inhibitor will be permanently attached to the enzyme and inhibit its entire enzymatic activity. So now we're going to talk about two types of reversible inhibitors or inhibition. Reversible inhibition, there are two types. One is called competitive inhibition. Competitive inhibition. Second one is non-competitive inhibition. So we're going to talk about both of them separately. Let's start with the competitive inhibition. In competitive inhibition, a competitive inhibitor, as the name is saying, competitive means they are competing with the substrate. So inhibitor is going to compete with the substrate. So that means whatever the structure or the shape of the substrate is, the inhibitor should have the same type of shape. So that's why they can compete with the active site, right? So competitive inhibitor has a structure that is similar to the shape or structure of the substrate, and it compete with the substrate for the active site, right? What does it mean? Let's say if I have an enzyme and you have a squarish substrate and this is a squarish inhibitor, so they have equal chances to go into the active site of the enzyme. So if the substrate comes first, is gonna bind to the active site and it converting into a product. But if the inhibitor wins and it binds to the active site first before the substrate bind, then the enzyme will become non-functional, because now enzyme bound to the inhibitor and there is no space for the substrate to bind to the active site, right? So that means, let's take an example. You have an enzyme. Now you have a competition between substrate and inhibitor. So if the substrate win, it's gonna form enzyme substrate complex, and now you're gonna get product, right? But if the inhibitor wins and it binds to the substrate first, again you're gonna make an enzyme inhibitor complex, and now you won't get anything, right? But this is a reversible. But how we can reverse this thing, how we can change this scenario? So as we know, this substrate and inhibitor both are competing it. So that means if I change the concentration of the substrate, means if I increase the concentration of the substrate, that means there will be higher chance of substrate to go to the active site. For example, let's go to this example again. On the right hand side corner, you have an enzyme, you have substrate inhibitor. Right now it has a 50-50 chance to get into the active site, right? But if I increase the substrate concentration, now what's going to happen? There is a higher chance of one of the substrate molecule out of the 5 to get into the active site compared to only one chance of the inhibitor to go into the active site. So if we increase the substrate concentration, we can increase the chance of substrate to go inside it and make a product, right? So you can reverse by increasing the substrate concentration. You can reverse that effect, right? Similarly, if you have a high concentration of inhibitor, then there is a high chance this enzyme inhibitor complex can form, and then the enzyme will be dead, right? So by changing the concentration of substrate, we can reverse the effect of inhibition. So this is an example. So you have normal like enzyme, you have substrate, enzyme go to the substrate, enzyme substrate complex. After the complex form, then enzyme does its function and convert substrate into a product. But if the enzyme is present, now you have a inhibitor is going to bind over here instead of substrate, and once this compound form enzyme inhibitor complex, then there won't be any product because the active site is already occupied by inhibitor. So now let's talk about the non-competitive inhibition. As the name is already saying, non-competitive means there is no competition between inhibitor and substrate, right? So in non-competitive inhibitor, inhibitor has a structure that is much different than the substrate. So that means structurally they are different, and the non-competitive inhibitor binds to an enzyme at a site other than the active site. What does it mean? If this is your enzyme with an active site, and then you have this is your substrate that's gonna go into the active site, and then this is your inhibitor. So this inhibitor doesn't compete for active site, right? It's gonna bind to the enzyme on any area other than active site. So let's assume it bind over here. So it's not binding to the active site, it's not competing for the active site, but it's gonna bind to the enzyme some other site other than the active site. But the time it bound to the enzyme, because of this binding, the shape of the active site is gonna change. So that means when it bound, now the shape of the active site is gonna be like this because of binding of inhibitor. Inhibit force the enzyme to change its active site, and if the active site shape is changed, now this substrate cannot be accommodated or go inside the active site because of the different shape of the substrate. So because of this changing in the shape of the active site, there is an inhibition of the enzyme, right? So it says that binding of enzyme at site other than active site and distort the shape of the enzyme by altering the shape of the active site. Now active site doesn't have the required shape for the substrate to bind, so because of this, it prevent the binding of the substrate, and it cannot have its effect reversed by adding more substrate. So can we reverse this reaction by adding more substrate? We cannot. Why? Because even though if we keep on increasing the substrate concentration, inhibitor is not going to compete with it. Why? Because substrate is not going to bind with on any other site except the active site where the inhibitor is not competing with. So by increasing the substrate concentration, there is no reversible because inhibitor is not competing for the active site. So there's a difference between competitive and non-competitive. So let's take an example. You have enzyme, you have substrate, normal reaction, enzyme bind to the substrate and convert into a product. But in non-inhibitor, if the inhibitor is present, it's gonna bind with somewhere else other than active site, is not going to the active site, right? So once it bound to an enzyme other than the active site, you can see the shape of the active site is now changed. Now the substrate cannot enter into the active site. If the substrate cannot enter into the active site, you won't get the product, right? So that's why if you increase the substrate concentration, let's say instead of 1s, if you would have 4s substrate, still it doesn't matter, because inhibitor is not binding to the active site, is binding to the other side. So it doesn't matter if you increase the substrate concentration or not for this non-competitive inhibition. Now let's talk about the irreversible inhibition. What is irreversible means? You cannot reverse. So irreversible inhibition is a molecule that causes the enzyme to lose its all activity, and is often toxic substance that destroy enzyme, means they are irreversible. Once it bound to the enzyme, this inhibitor, enzyme lose all its activity, it destroys, dead, and it usually form a covalent bond. So this kind of bond is very difficult to break. Once the bond is formed, the enzyme is completely non-active, right? It's not active anymore. So what are the example of irreversible inhibitor? Some nerve gas, some insecticide, or some antibiotic. There are a couple of example of irreversible inhibitor. So this is a table which normally compare between competitive, non-competitive, and irreversible, that we already talk about. So you can read it and compare all these things together at one slide. So now let's talk about one of the problem and then apply the concept that we learned. Identify each description as an inhibitor that is competitive and non-competitive. So you have to figure it out. Option A is saying increasing the substrate reverse inhibition. So we know its reverse is competitive, right? It binds to an enzyme surface but not on the active site. So that means non-competitive. Option C is its substructure is similar to that of the substrate. So if the structure is same, that means it's competing with the substrate, so it's competitive. Inhibition is not reversed by adding more substrate. So if you're not adding more substrate, that means the inhibitor should not be a competitive, it should be non-competitive. So this is how you normally analyze the problem and write the correct answer. So this is the answer already given for you that we already discussed in the previous slide.